in reality.
What are you doing December 3rd? What do you do?
It is very hot in here. You want to open the window?
Where are we confused? And you know, I when I go there in the summer, I had fish, so I was like,
This is going to pop
I
in.
should date.
Okay, you have a jacket.
Table Rock Lake has giant crawfish. So this summer,
That's
this summer, I'm taking a bunch of traps down there.
That's the day
I'm
that
doing
we're
crawfish on crawfish on crawfish.
That's a big deal.
Actually, I'm like right here in front of us. Like, ah.
Like I walked in with like a frozen bag of fish. I'm like,
I don't think I might stand back to this one.
those are comfort for that.
Maybe a little too much for that.
A window and a full
What?
length.
All right, let's get started. Today we are continuing with the introduction to the new case study. So, do you have any questions on what we went over last time? Part of the introduction that we're going through is to give you an introduction to case study, to actual content of the case study. The other reason that we're going through this introduction is to kind of give you a sense of how science operates in a broader sort of sense. So at the end of class last time, we had talked about, why would a species willingly eat another species that was toxic to it, to the point that in some cases you might die from eating it. And we were talking about the fact that Thamnophis garter snakes have genetically inherited prey preferences, and they oftentimes have very narrow prey preferences. So coastal populations of Thamnophis garter snakes will specialize on eating slugs, and inland populations will concentrate on eating other kinds of things. And they come to this predilection towards prey specialization because they find their prey by scent. And so they evolve scent receptors that are cued into certain types of scents and not cued into other types of scents. And it turns out that when you test snakes, you find that they have these dietary preferences as measured by the tongue flick rate. So whenever you present an item to a snake, it will start tongue flicking to smell that thing that you've presented to it. And if it's interested in it, it will continue tongue flicking at it so that it can know where that thing is that it wants to go and get. If it's not of interest to the snake, the tongue flick rate will actually decrease relatively quickly over time. And what Steve Arnold found was that if you cross these animals, if you take inland populations and cross them with individuals from coastal populations, the prey preferences actually change, indicating that this is actually a genetically inherited sort of trait. So the reason that snakes get tied into, having to eat a particular species is that it has these genetically ingrained preferences. And if the species starts evolving to get away from you in a sense, you have to just kind of be in it for the long haul. So we got to this point. As you read through this chapter, what are some of the kind of characteristics that scientists have as you went through this chapter? What things did you pick up on? How do scientists think? How do scientists behave? How do scientists do their work? Yeah.
They recognize what they don't know and consult people who do know that.
There's a book out called The Death of Expertise. Have any of you guys read it? It came out just the last couple of years. It's called The Death of Expertise. And it talks about how in American culture, we have grown to the point that we think that we can know everything just by going to the internet and Google it. And as a result, people who have expertise in areas just aren't respected and aren't listened to any longer. All one has to do is think about the pandemic to demonstrate that that is actually the case. Butch Brodie Jr. and Butch Brodie III, they both had particular sets of expertise. Butch Brodie Jr. studies predator-prey interactions. He is a specialist in salamanders and frogs and snakes and the predator-prey interactions of those types of organisms. His son specializes in quantitative genetics. That's what he has his PhD in. And he also worked on amphibians and reptiles also, but looking at genetic aspects of those things. As soon as they started getting away from those areas of expertise, they had to start drawing in other people who had other forms of expertise that they needed in order to continue to explore this particular case study. What else did you notice in this chapter? This speaks to the collaborative nature of science and how we don't typically see a bunch of single author papers any longer. And the reason is because you oftentimes need multiple areas of expertise in order to approach a project, which is why you guys are working in Greeks and Latin. What else did you notice? Yeah.
If they look back on creation to
Okay, so as you guys have been looking for older research in lab as you're writing your papers, you've probably relied on computer databases, but one of the problems with computer databases is they don't go back very far. They go back further now than they used to, but there was a time when no computer database for primary literature went back beyond 1982. And in a field of study like biology that's been going on for hundreds of years, there's tons of information that you're not gaining access to if you just limit yourself to research that you can pull off of an online database. Which is why we encourage you that when you're reading a paper, if it's a good paper about your topic, don't just get the information out of the paper, but go to the literature cited section of that paper and look at what those people cited and see, oh, this sounds like an interesting article that I should read because it pertains to my particular project. One of the quickest ways to gain entry into the literature is to find one good recent article and then mine that article for its literature cited. And then go and find those articles and see what those people cited, and then go and read those papers, etc. So you want to read old research because you don't want to reinvent the wheel, you want to do something that's already been done, but then it also informs the questions that you have about access to literature. What else did you notice about this chapter, which is not on regression yet? Where does Butch Brodie do only get his ideas? No, I mean, not in a very real sense in this case. Where did he get his original ideas? His father's previous work then informed his original questions, but where did he gain his original questions? Yeah, he was out in the field observing animals. He was a careful observer of nature, and as a result of that, he had questions about the things he was observing. Those questions then led him to remember this episode from his father's past that was informing those observations. But the first thing you have to do as a scientist, regardless of what field you're in, whether it's cell molecular biology, whether it's genetics, whether it's ecology and evolution, the first thing you have to do is you have to be a good observer, and you have to be curious about the stuff that you're interested in. If you're not curious, you're basically dead in the water in terms of being a scientist. You should go and, by all means, go and do something else with your life. You have to find a way of becoming curious about your field of study, and you have to find a way of maintaining that curiosity. What else? Yeah.
They were constantly altering their questions and like coming up with new ones.
Each time they came up with an answer to a particular question, that answer just led them to other questions. And so this iterative process that we talked about at the very beginning of the semester, the scientific method, you get to the end and you read reach the conclusion section, so you do observations, you have answers, or you have questions, you have hypotheses, you do experiments, you analyze the data, you get the answer, then there's an arrow that goes back up to the question section, and then you go through that process again. Science is an iterative process by which you bite off little chunks, you have questions about those little chunks, you find answers to those little chunks, and then you have more questions. You bite off those questions, you chew on those for a while, you get answers to those questions, which leads to more questions. Seldom do scientists make big earth-shattering discoveries. What most sciences do is you take the big block of wood that is, or the big rock that is our ignorance about the natural world and how it works, and you take a chisel to it and you chisel off your little sliver of that rock. And we make progress in science by basically pounding away at this rock until we've made this rock, incrementally smaller over time. Seldom are we taking this rock and splitting it in half and making really big discoveries that really clarify a lot of things. Oftentimes we're just whittling away at our own ignorance, and that is the very nature of science. And they illustrate this in this set of papers. What else did you learn about science and scientists in the process of doing this? What do these things illustrate about what you need to be a successful scientist?
Be really determined, willing to fail.
Yeah, when did they start working on this? Big Bush started back in the 1960s, then they picked this up in the 80s, and they've been working on this ever since, the 80s, 90s, 2000s, 2010s, 2020s. They've been working in the system since they picked it back up in the 80s for over 30 years. So one of the things you have to be is you have to be persistent. You have to be willing to dig into a problem and really dig into it in order to make progress on it. What are the characteristics of scientists in this chapter reveal? Well, obviously some of them are already on board. You need to be curious. If you're not curious, you're not going to have the persistence. You're going to give up relatively quickly because you're just not going to care about that set of questions. The Brodies are not particularly humble people. I know both of them. They're not particularly humble. You would never meet them at a meeting and walk away from a conversation with them going, wow, that guy's just really, really humble. But they do know their limitations, and everybody has limitations. Everybody has limitations. Dr. Judd went into way more detail on the light reactions than I think he should have. I would not have gone through that level of detail on the light reactions for a couple of reasons. One, it's not really necessary to understand light reactions to understand the difference between the C3 and C4 plants, because that has mostly to do with carbon fixation. Two, the light reactions are a lot of biochemistry. And I suck at biochemistry. Not only that, I don't like biochemistry. And so I recognize that biochemistry is one of my weak points. And so when I need to know about biochemistry, I either recruit the help of a biochemist or I run away from those topics as fast as I can. Because I too know what my limitations are. And sometimes there are things that I need expertise on. I was interested in population genetics a number of years ago. And so a guy was offering a course in Florida to learn about how to do genomic techniques for looking at population genetics, because I was interested in the genetics of a lizard species that I work on. So I enrolled in this course, and as a PhD, this was 2015, as a PhD person who's been out in the world doing his job for 15 years, I went and sat my ass down in a class in Florida for 2 weeks with a bunch of undergraduates, graduate students, and 1 or 2 postdocs, learning right alongside of them with, quite frankly, no more knowledge than they had. And in some cases, actually substantially less knowledge than they had. Because there to get better at what they do on a normal basis, and I was there to learn something that I don't normally do on a normal basis. So knowing what your limitations are is crucially important, and then not always just running away from them, but sometimes just taking the steps to become better educated about those things. What else do you gain from this chapter? About what it is to be a good scientist. So at each stage, they had more questions. After they figured out that tetrodotoxin was the substance that the newts are secreting, and they realized that the snakes were indeed resistant to this, they were interested in how tetrodotoxin actually works. At the molecular level, how does it actually work? And based on that, how are resistant snakes overcoming tetrodotoxin so that they can eat these newts? And then they were interested in, so you now have this ability, you can eat newts that are normally toxic to everything else, but almost all things come with a cost. And so what is the cost? What is the trade-off in having this ability? Before I leave that slide, the way in which tetrodotoxin acts requires that you know how nerves function. And to really get down to the nitty-gritty of it, it requires that you know about sodium channels and about the molecular structure of sodium channels. So for those of you who have been complaining that this is a course all about ecology and it's not about cell molecular biology and things that you would rather be interested in, guess what? We're about to start talking cell molecular biology. Guess what? You're going to hate it.
Sick.
The reason that you're going to hate it is because it's requiring you to shift gears somewhat in your thinking. The other reason that you're going to hate it is that it's actually just kind of complicated and difficult, in the same way that ecology has been complicated and difficult. And so we're going to shift gears, and inevitably, and I only say this because it happens every year. We go through the whole semester and students are like, man, I'm really not interested in evolution and ecology. We get to the last case study where we spend about half of the case study talking about evolution and ecology. We spend the other half of the case study talking about cell molecular biology, and they get to the end of it and they're like, yeah, I didn't like that. So just accept that when you're learning new things, it can be difficult and it can be challenging, regardless of how interested or not you're in it. Do you guys remember them talking about the geographical mosaic of coevolution? If you do, what was that referring to? Where do these newts live? Is there a map in the chapter showing the distribution of newts? It's a rhetorical question. There was a map. Where do they live?
The West Coast.
They live along the West Coast. They live in the Pacific Northwest, and they live, in California, about halfway down the length of California. So just south of San Francisco. Where do the snakes live? They also live on the West Coast, but where else do they live? These were so much more away, huh? So they live in Oregon and Washington and California also, but do they live other places? They live all the way over to the east. As a matter of fact, this same species that they're working on in this paper lives underneath my porch at my house. There's a hibernaculum of them. They come out every spring, and then they roam throughout the neighborhood, and then when they come back in the fall, they crawl back underneath my porch and spend the winter there, and it scares the crap out of people sometimes when they walk out and see a shrub next to my porch, and there are all these little snake heads sticking up out of it. What's that?
Do you enjoy the snake?
Yeah, sure. I mean, they're just doing their snaky thing. So you can think about these snake and newt populations where they co-occur as little geographic pockets, little populations in a sense. And in some of these populations, the snakes are really resistant and the newts are really toxic. In other populations, though, what happens?
Newts aren't as toxic as snakes, aren't as resistant.
The newts are not as toxic and the snakes are not as resistant. And so this is patchy. There are different places in the distribution where these two species overlap. And in some of those places, a lot of coevolution has happened such that the newt is really toxic and the snake is really resistant. In other patches, they find that the newt is really not very toxic, and as a result, the snake is not very resistant. And this has everything to do with just local evolutionary dynamics at these localized places around the range where these two species overlap. So there are these kind of hot spots for coevolution and cold spots for coevolution. Hot spots are where coevolution has been happening a lot, and cold spots are places where evolution has not been happening a lot. And so we'll see this anytime we look at broadly geographically distributed sorts of traits. Those traits will vary with geography, and when you have coevolution, that variation can be actually quite striking in some cases. So when they got to the end of this set of papers, they actually wanted to quantify how consistent is this toxicity in the newts and how consistent is the resistance of the snakes as we look across this broad geographic area. And in the last paper that we'll talk about, you'll see they found some very interesting findings that are, once again, based in the molecular biology of how these snakes attain resistance and the genetics of how these snakes attain resistance. Questions about this overview? We're going to read 4 papers. The first 2 papers are more kind of ecological in that they look at whole body performance of snakes when they give them different levels of tetrodotoxin. The last 2 papers are very molecular when they start to look at what is the mechanism by which tetrodotoxin is toxic to snakes and what are the mechanisms by which snakes have evolved resistance. And that's all a bunch of molecular biology. So you're going to get both fields here at the end. All right. What do we always do when we start on a new case study?
Go over the measurements they used, or like their process of how they got their results.
Yeah, we introduce you to any statistical methods that they use frequently in the papers that you're going to be reading, so that you, when you go to write your literature summary, you know kind of what's going on with those things. Once again, we're never going to ask you to do a regression, to perform a regression, but we want you to be well enough informed about what a regression is that you can actually understand what they're talking about. Not to mention, regression is like the second most common thing that biologists ever do in life. The most common thing they do is ANOVAs, the second most common thing that they do is regression. And so it's also just generally a good thing to know. So we'll run through that real quick. So regression is just when we estimate the relationship between one continuous variable and another continuous variable. And in almost all cases, we express this in the form of a line. Can somebody remind us all of what the equation for a line is? Okay, y equals an x plus b, good. What is the m in this?
Slope.
It's the slope. What is the b? It's the y-intercept. If x is the parental trait, parental phenotype, and y is the average offspring phenotype, from those families. What is M? Test 2 material. It's heritability. So we can also use this equation to estimate the heritability. And this is precisely the way in which we measure heritability in a quantitative genetic trait, which is exactly the kind of trait that little Butch-Brody III, actually works on, because he's a quantitative geneticist. That is not, however, how we're going to talk to you about regression here, but just put that in your, get that stuff back in your head for this case study as well. Correlation is somewhat different in that correlation measures the degree to which as one thing varies, the other thing varies. It's a subtle distinction, but an important one in terms of kind of how you interpret correlations versus regressions. But generally we have y equals mx plus b. In this case it's y equals bx plus a. It doesn't really matter what those variables are. You just need to remember that the thing that you're multiplying times the x variable is the slope, and the other thing is the y-intercept. It doesn't really matter. Could have used q and r in the equation. So there are two types of regression. Only one we're going to talk about in any depth. The two types of regression are a model 1 type of regression and a model 2 type of regression. A model 1 regression is a situation where the independent variable is something that the researcher sets themselves, and it can be measured without error. And so the x's are fixed. You say, this is x, and I want to see how y responds to me setting x this way. Don't worry about what the expected value is. It's just the line of the equation. But basically, for any given of x, y is independently free to move around. It's independent of all the other data points, and it's normally distributed in terms of its variation around the line. And those are similar to the assumptions that you make when you make an ANOVA. It's just that they're assumptions for a regression. So most people will analyze their data this way, even if they had absolutely no control over the x variable. Let me say that again. People always analyze their data this way, even when they have absolutely no control over the x variable. It's just the way it is. And you can write journal articles trying to convince them that they're doing it the wrong way, and guess what? They won't listen to you. They're going to keep on doing it the same way that they've always done it. There is another way that is not ordinary least squares regression for dealing with situations where the x variable and the y variable are free to vary. They're both what we call random variables. This is called a standard major axis regression. It's also sometimes called a geometric mean regression. It's easy enough to do. Standard statistical packages will do it, but scientists are so ingrained in their ways of doing things that they just generally don't do it. Dr. Judd is
Yeah.
wrinkling his
We've talked
forehead.
about this several times, and I just realized that there was a method that I was going to use for analyzing some phylogenetic data using a modification of the ordinary least squares method, but really it should be a modification of type 2, not type
Yeah, because
1.
you don't have control of either
Yeah.
of any of the variables. Yeah, it's
So I just noticed this embedded deep in another subfield, this problem he just mentioned.
yeah, and I learned about geographic mean regression when I was in graduate school, and it's the right way to do things, but nobody ever does it, so I'm not going to impose upon you. I'm just going to make you aware of it. But from here on out, you can do what all the other biologists do, which is just ignore this section of the presentation. So, ordinary least squares regression. In this case, I'm going to give you an example that is an actual ordinary least squares regression problem. Because what the investigators did was they set the relative humidity in chambers where they were growing flower beetles. And then they looked to see how much weight those flower beetles lost over a period of time. So in this case, they are actually setting what the x variable is and then seeing how weight loss is affected. So the nature of regression is you set x and then you look and see how does y respond. So it's a very causal analysis. rather than a correlation where you just see how these two things covary. One thing may be driving the other thing, the second thing may be driving the first thing, or these two things could just be correlated with some third thing that you didn't even measure. And so there's no causal relationship there. In this case, there is a causal relationship. I, the scientist, changed relative humidity, and then I looked to see how water loss was affected. They weighed these beetles in 9 groups of 25 beetles. They got the average weight loss or gain after 6 days. And the independent variable here is relative humidity. The researchers set that. And the dependent variable is weight loss. And this is what the data look like. So when I first saw these data, I looked at them and I'm like, that doesn't look like a researcher set this, because who would set the relative humidity at 29.5%? That's ridiculous. Why didn't they just set it at 30%? Then I went and I actually looked up the actual study, and they did set it. They just weren't very good at setting it. But when you look at these data, what do you notice about the pattern? Yeah, there seems to be a trend. At low relative humidity, you tend to have larger weight loss, and at high relative humidity, you have less weight loss. And so the data end up looking like this, and that's exactly what you see. You see this trend. Now, the average weight loss, the mean of the Y variable, weight loss, is 6.022 milligrams in this case. And so we can actually just draw a line here showing where that average is. So this is the average value of the Y variable. And you can see that in some cases these data points are quite far away from that average value, and in other cases they're quite close. What do we call the distance between a mean and an actual data point? There's a term for it that we gave back when we covered ANOVA. Anybody remember? Give me a letter. A.
E.
Oh. Oh.
T.
Hmm?
S.
Do you know what it is, Trevor?
No.
It's the distance between the average value and a data point. Sometimes also referred to as error, but in R, when you do it and you calculate it in ANOVA, it'll give you the sum squared of the treatment, and it will give you the sum squared of this.
Residual.
Yes, very good.
Hey, she's a G. Give him
Nice.
a point. Give him a point.
So the difference between here and here is a residual. The difference between here and here is a residual. Residual, 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 residuals. Now, there's a pattern to the residuals, isn't there? Where are the residuals small? They're around the middle of the x variable. And what happens as you move to smaller x variable values? The residuals are bigger and they're positive, and as you move to larger x variable values, they get bigger and they are negative. And so these residuals have a pattern, and the reason for this is y equals mx plus b, where m is a slope of 0. It's a flat line. It's a horizontal line. And the reason that there's this pattern in the residuals is because the best fit line isn't a horizontal line. What is the best fit line going to look like?
What was the question? Sorry, I apologize.
This is not the best fit line. This is a slope of 0 line. This is the null hypothesis, in a sense, that x has no relationship to y. That's not the best fit line. What would the best fit line look like? Jamie? You look like you were drawing something. Yeah, it would look like this. It would look something more like a downward sloping line where the slope is actually a negative slope rather than a positive slope or a slope of 0, right? Yeah. See, I knew you were thinking it because I saw your hands moving in space. So if we actually do, oh man, I was pretty damn good. If we actually do the regression, what we find is that the best fit of the line is actually something that has a negative slope. It's a really small negative slope, but it's a negative slope. The y-intercept is up here at 8.7, close to 9. And the r-square value, which we'll talk about in a few minutes, just tells you that the data points are clustered really closely to the line. But what has happened to the size of the residuals? All of the residuals have become smaller. Is there now a pattern to the residuals where some parts of the graph have big residuals and other parts of the graph have small residuals? No, they're all just kind of the same. And so this is the best fit line. The best fit line is the line with the slope through the data at whatever the average relative humidity is. You could basically put this line on a teeter-totter thing here, and you move that slope around until you get to a slope that is the slope with the smallest value for the residuals. And so we can calculate residuals the same way. It's the difference between the value and the running mean. One of the ways of thinking about the regression line itself, the regression line itself is the average value of y as x changes. So normally when we're doing an ANOVA, the residual is the actual value minus the mean squared. That's the residual value. This is what we use when we're calculating variance. Remember, we sum all these up, we divide it by the number of observations, and that's sample variance. Well, in the same way, if the line is the average value of y as x is changing, then this is simply y minus y bar. This is y minus y bar. This is y minus y bar. Y minus y bar, y minus y bar, y minus y bar, y minus y bar, et cetera, et cetera, et cetera. We can square those up and we can add them all up and we get that aggregate amount of variation around the line. And what regression does is it minimizes what that variation is. And that's where you get the best fit line. There are relatively uncomplicated formulas for doing this. Once again, you don't need to know those. You just need to understand what regression is doing, and you, in particular, need to remember what a residual is. These guys, in their papers, they use residuals a lot. So if you don't remember what a residual is, you're going to be really lost when you go to read these papers. Any questions about this up to this point? Okay, these residuals are now smaller. R square is less important. R square, though, don't confuse R square with anything having to do with statistical significance. R square just refers to how variable the data points are around the line. In this case, each of the data points falls on this line, such that if you know what x is, you know what y is. So in this case, x explains 100% of the variation in the y variable. If you know what x is, you know what y is. That's an r square of 1. It's 1 because it's 100% of the variation. In this case, if you know something about x, do you know something about y? Yeah, do you know quite a lot about y? Yeah, sure. In this case, x explains 90.37% of the variation in the y variable. Because the r squared is 0.9037. If you know x in this case, do you still know something about y? Do you know as much about y as you knew up here? No, because x is doing a worse job of predicting what's happening with y, to the point that x only explains about 70% of the variation in y. Do you still know something about y if you know x in this situation? Do you know as much about y by knowing x as you do in this situation? No. For one thing, it doesn't look right. It doesn't look like you know much about y. But then the other thing is the r squared tells you that you don't know as much about y when you look at x. X only explains about 44% of the variation in y in this case. So separate r squared from a test of significance. R squared is just a measure of how much variability in the response variable is explained by the variable that you set, in this case the x variable. Here's a graph from an actual paper, shockingly enough. These are, you guys know what barn swallows are? It's these birds that hang out at bridges. You oftentimes see them at bridges and they're spinning around in air. They have a long forked tail. The forked tail actually is a sexual signal that males use to advertise to females, and females assess males on the basis of their tail length. If you have a long tail, you're really sexy. If you have a short tail, you're not particularly sexy. And this person was looking at the relationship between male tail length and female tail length to see if females actually tended to choose males with longer tails, and if they chose males that were more like themselves in terms of tail length. And what he found was that females with short tails tended to mate with males with short tails, and females with long tails tended to mate with males of long tails. But what I want you to do is look down here in the caption, because the caption is where you get important information. And for this top graph, the equation is female tail length is equal to 68.96 plus 0.17 times male tail length. This is a significant regression. The F value is really huge. The P value is really, really small. It's less than 0.0001. And the R square value is 0.09. Okay? So, what do we do with all this information? This part right here is what? Part of the line. It's the slope. What that tells us is that the slope is positive. The p-value tells us that that slope is different from a slope of 0. It's not tons different, but it's different enough that you can detect. What does R squared tell you? Okay, so that's one way of thinking about it. What's another way of thinking about it? Sorry, I heard a couple of things.
How much x explains y?
Yeah. How much does knowing something about x tell you about variation in y? Does x tell you a lot about y in this case? No, it doesn't, does it? It tells you almost nothing about y. It tells you 9% about y. X explains 9% of the variation in y in this case. So then the question is, well, that's not a terribly useful regression. You want a regression that is explanatory. You want a regression that tells you that x is related to y in a meaningful way. So my question to you is, why is this regression that doesn't do much in terms of male tail length variation, explaining variation in female tail length, why is it a significant correlation, or a significant regression rather, statistically. What have I not told you about the F statistic that we always want to know? And we usually put in parentheses next to the F. In this case, these numbers are 1 and 336. Do you guys remember what these are from doing an ANOVA? They're the degrees of freedom. 1 degree of freedom is used up in estimating what the slope value is, and the rest of the degrees of freedom are available for testing the hypothesis that this is a slope different from a slope of 0. So the sample size in this case is 338. So they measured 338 pairs of birds in this analysis. What do you know about the relationship between sample size and your ability to find a difference? In this case, the difference between a slope of 0 and something else. The bigger the sample size, the easier it is to find a difference. They have a gigantic sample size, so they can actually detect differences of slope that are really, really tiny, even though that regression line might not tell you hardly anything about what's happening with female tail length as it's related to male tail length. And this is one of the tricky things about regression. There is such a thing as actually having too much data in a regression, because you can find little tiny differences that aren't actually very biologically significant. You were about to say something, Dr.
I'm just imagining if you had done a preliminary study and you had used Dr. Kowinski's rule of thumb on how many pairs to include in your initial sample, how many pairs would you have probably measured? You guys remember what that rule of thumb was for sample size?
What's closer to infinity than it is to 0? The lichen group knows this, because this is how many trees they went and measured. The lichen group knows this, because this is how many trees they went and measured.
Yeah, it's,
Jamie, how many trees did you guys measure?
We measured 30.
You measured 30. Why? Because 30 is way closer to infinity than it is to 0 in terms of sample size. You don't really gain much in statistical power once you get past 30.
So if you had done this preliminary analysis and just picked 30 pairs, do you think you would have found a relationship? Would you have been able to detect a significant relationship?
Yes.
If you had a 1/10 the sample size?
I don't know, you just said 30 is close to infinity. Sounded
Sure,
right.
but that's just why we use 30 as a good sample size to start a study with.
Oh.
This is way overkill for detecting a difference in this slope. If you think that the slope is actually like
Meaning.
worth paying attention to.
Hey, the more the better.
No, you don't need nearly that much.
The more the merrier. That's how I think about it.
At a party, maybe, but not
No,
necessarily. for
eating
regression.
more food, the merrier. Everything. More is better.
More is not better.
No.
When is more not better? What?
More diseases that are affecting your body.
More cures.
When it costs you money to gather more data that you don't really need because it doesn't really shed any more light on your answer, then yeah, more
Do
is
you get
not
more
better.
money?
No, you don't get more money. You get the money that you get.
Well, then you use to get that.
Well,
And you should
so-
use it to get 30 rather than 300.
I always just get more money. Get your money up. Get more money and be happy, see?
But do you, just to be clear though, do you all understand the point here is that if this is the R squared that we're really gonna find, that if you only had 30 out of these 338 points, would you have the power to detect a significant difference from 0 for the slope? Probably not, right?
If you had only 30 data points, this is maybe a little clearer. If you had only 30 data points, this is probably not gonna change.
Right.
What would change is
Yeah.
this would probably not be significant anymore. And then you would look at this and you would say, oh, it's not significant, which is why it doesn't really explain much of the variation going on in X and Y. And so in this case, there's a p-value that doesn't really match up with what the r-square value is telling you, because the regression is significant, but it doesn't really explain much of the relationship between female tail length and male tail length in this case. So it's just one of those situations where it's so much statistical overkill that it gives you the impression that this is a relationship that's actually meaningful that actually probably isn't meaningful at all. And so it's one of these situations where we can discuss the difference between statistical significance versus biological significance. And this is a situation where you have a statistical significance that probably doesn't mean that much to birds flying around the bridge somewhere. And that's the point. All right. We've already seen some data that we've looked at with regression. This is the regression of height data that we talked about back when we were talking about quantitative genetics. Once again, the x variable is not something that is set by the researchers. It's something that the researchers just observed. But as x varies, y also varies. And in this case, the slope of the line is 0.9165, which also happens to be the heritability for height for this particular sample. So we can use regression to measure heritability. We can use regression for all kinds of things. What's the relationship between a person's height and their shoe size? It's a stupid, trivial sort of data set, but it's a big data set. It's 408 data points. And you can ask the question, do men and women have different shoe sizes? Well, because height is correlated with shoe size, we would normally use an ANOVA to analyze this data, but you can't really use an ANOVA to analyze the data because when you look at it, there's a big difference in shoe size between females and males. This is the ANOVA table down here, the degrees of freedom, and we have the p-value, which is very, very low, indicating that there's a big difference. But when we look at the data, we see that there's a cluster of males up here, there's a cluster of females down here, and so shoe size in males is bigger, partly because males are also taller. And we can jitter the data so you can actually see the individual data points. But once again, you see that there's this cloud of male data, and then there is a cloud of female data. And so part of the difference in shoe size is actually due to differences in the height of the individuals. So one of the ways that you can look at these data, and it's not the best way, but it's one way to look at these data, is you can ask yourself, for individuals that are 75 inches tall, some of these individuals are above the line, meaning they have positive residual values. Some of these individuals are below the line, indicating that they have negative residual value. So these are negative residuals, these are positive residuals. We could then record that for every individual in this data set, and we could get the residual value, which basically says, at a given height, for a given height, do you tend to have big feet or small feet? At a given height, do you tend to have big feet or small feet? At a given height, do you tend to have big feet or small feet? What do you notice down here where there's a mixture of males and females? So here at 65 inches tall, you see that there's a mixture of males and females. Where are the females? They're both above the line and below the line. They have positive residuals and negative residuals. Where are all the dudes? They're all positive residuals. There's 1, 2, 3 dudes here. They're all positive residuals. Here at this point, you have a bunch of males and females. Females are both what? Positive and negative residuals. What are the males? The males are all positive residuals. And so one of the other ways that you can look to see if there's this difference is by actually performing a regression, getting the residual values, and then analyzing the residual values, because presumably the residual values are removing statistically the effect of changes in differences in height. And if you do this analysis, where you calculate the residuals and then calculate the residual shoe size, you find that they're still different, but they're not nearly as different as they were before when you were just looking at the real data. And that's because you've removed this correlation, this regression between shoe size and height. This statistical removal of a covariate is something that they do repeatedly in the papers that you will read. And so they will talk about residual resistance. And they talk about residual resistance because resistance varies with body size. How many of you ever tried to poison someone? Did you?
Yeah.
Jamie, did he try and poison you? No. You haven't tried to poison anyone?
No.
If you were to poison someone, what should you make sure you do? Get enough into them that they actually die. So what is it good to know?
Wait.
How much they weigh. Because if we inject the same amount of poison into David versus Adeline, they're gonna, if we inject them with the same amount of poison, David's going to survive, Adeline's going to die.
David built it.
What? What was that?
I said David was just built different.
Yeah, you're just heavier. And so if you're gonna kill somebody with a poison, you wanna know what their weight is so that you give them an appropriate amount of poison so that you actually get the job done. Yeah,
This could also be an example, like, if you want to cure someone,
sure.
and, you know.
Well, that's the less interesting way. So you know that they've started giving COVID vaccine doses to children now, right?
Yes.
What's different about those doses? Yeah, they're a fraction of the amount of what they give to an adult, once
Really?
again, because of this effective size. So you want to remove the size of the individual from your measure of resistance, because larger animals can absorb more of a toxin before it starts to affect them physiologically. And so you can do a regression between how much it takes to knock you back with size as the X variable. You can remove the variation in how much toxin it requires to slow you down a certain amount. And you can statistically remove that by looking at the residual values.
Some
And
of
so-
you, if you're in the medical field, might have to do this with anesthesia or something
That's
someday.
what
Yeah,
I'm saying.
they take
Saving
your-
lives, not cure.
They take your weight right before you go into surgery because the anesthesiologist needs to know how much you weigh so that they know how much to dose you to get you under while not killing you.
Killing you.
Yes. Sorry, I tend to go to death.
Yeah, death is cool, like, it's part of life, but.
My sister is really into true crime. Like, she knows a number, she's a writer and she knows a number of true crime writers. And she's in this writing group that writes true crime, and so I always just go to death. It's Teresa's fault.
So like, if you want to kill someone, if you put one of these newts like in their food,
Yes, I can.
then they just die.
But depends on population once again because of the geographic mosaic.
All right, one of these really poisonous
evolution.
ones, like, could they track you, like, realistically?
Oh yeah, so I think I mentioned last time that before this case study became kind of a textbook case study in evolution textbooks, nobody knew about tetrodotoxin unless you like went to the University of Texas at Arlington where these people worked on this system. But since it's gained traction and it's now like, it literally is a textbook example of predator-prey coevolution, it's become much more widely known. And as a result, 30 years ago on TV crime procedurals, no one ever died by being dosed with tetrodotoxin. And now it's fairly regular on TV crime procedurals that the murder weapon has been
So like real
tetrodotoxin.
life, like if you wanted to kill someone.
You don't kill anyone.
No, I mean, obviously that, but like, do you think you'd get away with it?
No, because it's so much better known as a toxin.
They would
Yeah.
look for their killer.
Yeah, they would find you because.
They would find me, but.
No, because they would come to me if it was a murder around here. They would say, "Oh, this person died as a result of tetrodotoxin poisoning. Who are the students in your evolution and ecology class?" We would give them your names and we would tell them to pay special attention to Sam.
Wait, do they do that? They ask people.
Cops come to
Yeah.
professors. Cops
Watch
come to
crime shows
professors.
on TV.
Not at all. I don't watch TV.
All right.
Never seen Lord of the Rings either.
So,
All right.
in one of the papers, they show you a figure like this. And what it shows you is it shows you the speed that the snake is running at before you inject it with tetrodotoxin. And then they show you the speed after they've injected it with tetrodotoxin. So it stands to reason that the faster you are before you get injected with tetrodotoxin, the faster you might be after you're injected with tetrodotoxin. And so you need to remove the effect of initial baseline speed. So this is an animation. So I noticed that some of you download the PowerPoints as a PDF or something like that. This is one situation where you won't want to do that because the animation works to make the point. So if you are fast before you're injected and you're fast after you're injected, there should be a positive correlation. So we can think about the effect of tetrodotoxin on snakes by asking ourselves, what would the situation be if there's no effect of tetrodotoxin and there's no trade-off? There's nothing to be gained. Well, if there's no effect, you're just as fast after injection as you are before injection. And so tetrodotoxin isn't affecting you, and so there's just this one-to-one line. We can then say, what would this line look like if there was an effect of tetrodotoxin, but it affected all snakes equally? It affected slow snakes the same way that it affected fast snakes, in that it slowed them down. What would this line look like? Tetrodotoxin slows you down because it messes with your nervous system, but it slows down all snakes, slow snakes and fast snakes equally. What would this line look like? What? So it would be, so what would happen? Would it be higher? Would it be lower? Would the slope change?
Mine would be flipped.
Flipped. What do you mean by flipped?
It would like have a downward decrease.
No, that would mean that slow snakes are faster after getting injected and fast snakes are slower after getting injected. So it wouldn't be flipped. That would indicate that there's a trade-off. And a trade-off is so severe that actually slow snakes get faster after being injected with tetrodotoxin. It wouldn't be less steep because it would affect all snakes equally. What would happen to the line? It wouldn't be horizontal. Hmm? Not the x-intercept, the y-intercept would change. So just the y-intercept would change. You take snakes, you measure their baseline speed, then you inject them with tetrodotoxin. It slows everybody down, but it slows everybody down equally. That's what you were getting at.
Yeah, the x-intercept also changes.
What's that?
The x-intercept also changes.
Well, it does also change, but we don't normally think of
Yeah,
x-intercepts.
it's
It's not a part of the equation of the line. Yes, it does. But the bad thing is the x-intercept actually gets bigger, and so that's a little counterintuitive if we think of snakes slowing down. So in this case, faster snakes are still faster after tetrodotoxin, but faster snakes aren't any faster or slower relative to other snakes as they were before because there's no trade-off. So this is what the line would be like if there was an effective tetrodotoxin, but there was no trade-off. So when we actually go and gather the data, what we find is that the best fit line through this data set, if we look at the average baseline speed and the average post-injection speed, we can put a line through there that is the same slope as this one, which is the effective tetrodotoxin with no trade-off, but at the y-intercept of this data set. Is that a good fit of that line to those data points? It isn't. We have a lot of negative residuals here. We have a lot of positive residuals here. So what does this line need to do in terms of its tilt, in terms of its slope? Yeah, we need to take that slope and we need to cant it so that it looks like this. This is now the best fit slope through the line. And what that means is that this slope is lower than this slope such that tetrodotoxin affects faster snakes more than it affects slower snakes. Slower snakes are actually more resistant to tetrodotoxin than faster snakes are. if you're fast at baseline speed, you're going to take a bigger hit to post-injection speed than if you are a slow snake at baseline speed. That affects your post-injection speed less. And so when you get rid of all the other figures, you're left with this figure where you see the effect of tetrodotoxin without a trade-off versus the effect of tetrodotoxin with a trade-off. This is the one-to-one slope line. This is the effect of the trade-off. And when you look at these figures, the dotted line is the one-to-one slope. The solid line is the slope that actually fits the data points best. So when you go to read these papers, keep this animation in mind because this animation explains how you can interpret this particular figure.
You definitely want to remember this because notice when you just look at the picture, the dotted lines don't look the same. That's because the axes are different in each plot. So just watch that.
Questions about this? This figure will just look like this when you actually open it up in a PDF. If you open it up in PowerPoint, you can run through the animation and it will make hopefully more sense to you. So keep in mind the idea of what a residual is and the fact that they're going to be using regression a lot. So just embrace regression. If you have any questions about regression, think about it over the Thanksgiving weekend and come in and see Dr. Judd or myself and we can help you out with it. Dr. Judd has your
I have VNL summaries for most of you. There are a handful of you that I don't have them, but you'll get them via email this afternoon. So we've got a couple of stacks here.
Oh, yeah. Max. Oh, yeah.
I've never seen.
Amanda.
I think I might have seen it before.
Emily.
Dr. Judd, I'm so excited to meet you. I was talking to you and my
Okay.
Right here.
all I have is the conclusion.
Oh, yeah.
Thank you. It's okay, I used to sit back there.
You used to sit back there.
Yeah.
I'll,
Half of this grade is based on the quality of your review, and half of the grade is based on Dr. Judd's assessment of your paper.
Yeah. So whatever the number is, that's half, and then we add to it for the other half, the grade on the review.